Good morning, everyone. Myself, Devika Rani H K research scholar from Tumkur University, Tumkur. I am pursuing my PhD under Dr. Parimala B, Associate Professor, Department of Zoology, Tumkur University, Tumkur. Today, I am here to explain about toxicological effects of sublethal isoproteron on neurobehavioral and morphological endpoints in Cyprinus scorpio. Next, moving on to introduction. Nowadays, agricultural chemicals are widely used to clear unwanted weeds in the agricultural crops. These agricultural chemicals are widely used in the agricultural fields to get more yields by destroying or to clear the unwanted weeds which are grown in the agricultural fields. These agricultural chemicals, upon using in the agricultural fields, may result in the increase in environmental pollutions, and this causes the toxic effects on water bodies and also on aquatic organisms by entering the water bodies. Mainly, the fishes absorb via gills, skins, or GI tracts, which are rapidly metabolized and excreted and bioconcentrated in various tissues of the fishes. These accumulated agricultural chemicals results in the death or long-term damages to the fishes. Next, moving on to the methodology, where the test chemical I used in my experiment is isoproteron. This isoproteron is the herbicide, where this herbicide, isoproteron herbicide, is belongs to phenylurea group. This isoproteron herbicide mainly used in agricultural crops like wheat, sugarcane, citrus fruits, cereals, etc. The main usage of this isoproteron in the agricultural fields to remove the unwanted weeds in the agricultural crop area, and also to remove the broad leaves which are grown in the crop fields. This isoproteron can harmful to aquatic environment it not disposed correctly, and the surfactants are recognized the ability to reduce the water surface tensions and thereby improving the solubility of the different contaminants. Where the test model, in my experiments I used up the test model as the Cyprinus scorpio, and this Cyprinus scorpio belongs to a family Cyprinidae. These are my laboratory setup. This figure 1 shows the fisheries collections, and figure 2, figure 3, and figure 4 shows the experimental setup in my laboratory. Next, moving on to the objectives, identifying LC50 values to determine the concentration of the isoproteron that results in a 50% mortality rate in Cyprinus scorpio through systematic exposure trials. Next objective is to analyze the behavioral changes and to monitor and record the behavioral changes in the Cyprinus scorpio exposed to varying concentrations of isoproteron and establish concentrations between these behavioral changes and the biochemical obtained. And next objective is to examine the neural toxicity to monitor and record the ACH and ACHE in the Cyprinus scorpio exposed to varying concentration of the isoproteron. Next, moving on to behavioral studies. In behavioral changes in the fishes were observed immediately after the applying the test substances into the experimental depths, and this shows at the lowest concentration of the test substances shows the normal little bit changes in the fish behavioral activities like erratic swimming pattern, loss of balance, they just laterally laying on the water, and they just rapid opercular movements. While the concentration goes on increasing, at the highest concentration they shows severe loss of balance, lying lateral or at the bottom, and rapid ocular movements with the opened mouth, and the ocular disorders also found in the chemical exposed fishes. But in controlled group they exhibit normal behavior, and they just behave normally in the control group. Next, moving on to the morphological studies, where this figures shows how the chemical exposed fishes shows the different disorders. Like first figure shows that caudal bending. In the figure 1 show caudal bending, the exposed fishes shows their body is in normal, they just in the normal shape, but in the caudal bending. Next is the figure 2, ocular disorder and open mouth. This open mouth and ocular eyeball shows darkened regions, and this figure 3 shows the hemorrhages. This shows the clotting of blood at the dorsal side of the fish bodies in the chemical exposed fishes. Next is the neurotoxic studies. Behavioral abnormalities including erratic and rapid swimming, heightened the activities, loss of balance, and increased bottom dwelling behavior. Observable morphological alterations including the caudal curvature, changes in the body pigmentation, and ocular disformities. This all affects by likely associated with the acetylcholine esterase inhibition, resulting in the accumulation of acetylcholine in the synaptic junctions and system subsequent to overstimulations of neuromuscular systems, leading to impaired neural signaling. This graph shows how the ACHE content in the brain tissues, and how uh, this ACH accumulated in the synaptic junction while varying the concentrations. Next is moving on to conclusion. Sublethal exposure of the isoproteron caused significant behavioral and morphological alterations in Sepinoscorpio fingerlings, indicating strong neurotoxic effects even at the lower concentration. The persistence of these impairments at the recovery period highlighted the limited physiological recovery and potential long-term damage, and these findings emphasizes the ecological risk of isoproteron contamination in the freshwater ecosystem and need for strict regulation of continuous environmental monitoring. These are some of the references I used upon my experiments. Thank you.